Hi, everyone. It's Mr. Vallejo. Thanks for coming to biology class today. Today, we'll be taking a look at structure of DNA. And if you take a look at there's a reminder for you to uh, take your notes and in your Cornell template, or if you want to write it on a piece of paper and set up your Cornell notes this way, it'd be awesome. Let's take a look. Structure of DNA. DNA structure is the same in all organisms, whether it's a eukaryotic organism or a prokaryotic organism. That is to say, it's an organism with a nucleus or it is a bacterium. They both have DNA. So DNA structure is the same in all organisms. This has been known for a little while now. First, we have Miescher, who discovered nucleic acids in the late 1800s. This actually then is, it's only been, 150 years when we're starting to figure out what the structure of DNA is and figuring out that it is actually DNA, and they thought it was proteins. That was the device where we took the genetic information and shared that with the next organism or pass the genetic information along. Well, how did we do that? Well, this is Griffin's experiment. In Griffin's experiment, there's a lot of text here, but basically what he did was he took some pneumococci. This is the bacterium that causes pneumonia, and there are two different strains of that bacterium, and the one that says R is a non-pathogenic strain. It does not cause the disease. It's called the R strain because the R stands for rough. And if you look at it under the microscope, because it doesn't have a capsule, the cell membrane looks a little jagged. So that's a rough strain. Whereas if it has a capsule, and that's called the S strain, and the S strain does cause a disease. And that makes sense because if it has a capsule, then that bacterium is more likely to exist and be alive and stay. And that is what is happening there. So if you take a look at the bottom left of my slide, it says live R. If you take some live R strain bacteria and you inject that into a mouse, well, it still lives. Well, that makes sense because you injected bacteria in there, but the R strain doesn't cause a disease. On the other hand, if you take that bacteria, the S strain of that bacteria, and inject it into the bacterium, well, shoot, they die. And that is that is unfortunate, but it's what is to be expected. So we need to do that to check to make sure that was actually the case, like you see in the mouse on the photograph on the right. That mouse was injected with the S strain, but actually, if you read the inset underneath the photo, it says a mouse injected with both heat-killed S strain and live non-virulent R strain dies. Well, before that, what we can do is take the bacteria that cause the disease, the S strain, and we kill it with heat, and then inject that into the mouse. Well, the mice still live. And that makes sense also. Even though the S strain causes the disease, they have been killed. And so since they're dead, they shouldn't cause the disease and these mice live. Well, here's another thing. If you have live R strains that don't kill the mice, and you have heat-killed S that don't kill the mice, they should live. And that's true for almost all of them. I wrote here mostly. But as you can see on the photo here, wait a second, there are a few that die. Well, what happened there? Some of them die. Well, they die because there's something that's transferred from the heat-killed S bacteria to the live R bacteria. What is that something? Well, that was the question for a while. So then some other experimenters named Hershey and Chase did an experiment with bacteria. And this is called a bacteriophage. phage means eater, and bacteria. So we have a bacteria eating that virus. And this famous experiment by Hershey Chase was called the blender experiment. And what they did was they grew the virus in such a way that the DNA was replicated in the presence of a radioactive phosphorus. And over here, you can see that these viruses were grown in radioactive sulfur. Well, sulfur is found in the protein coat of the virus, whereas
the owner of
here, these green ones right here,
plate 8, V as in Victor,
that's not,
NLNOH.
So you have,
there were some car keys found behind it. The keys will be located in the counseling office.
Anyways,
white
you have the radioactive phosphorus and the radioactive sulfur in these two different types of viruses. But if you inject them and follow them, and you follow the radioactivity, well, here's what could have happened over here. Let's say, all right. So this is the DNA, and this is the protein. Well, the protein, after the virus attacks the bacterium and the bacteria go on, well, guess what? There's no more radioactivity in here after you shed off the virus. How do you do that? With a blender. And it's spelled with a O because this was a British experiment. You know how they sometimes spell things a little bit differently in England. So here they put this in the blender and they cleaved this off and the DNA went in. So now you have the cell that has the virus DNA in there, and then this bacterium starts to make viruses. But there's no radioactive stuff in these viruses over here. On the other hand, here you have a radioactive phosphorus in the DNA, and that DNA then that virus was allowed to attack the bacteria. They put this in a blender, and then the heads came off. Here's the head. But if the radioactivity is in the DNA, that DNA should still be radioactive. And there it is right there. That's what happened. So over here, we saw that there's something that's transferred from the heat-killed S to the live R, but we don't know what it is. They thought it was protein for a long time, but this experiment shows that no, it's not protein. Protein, the radioactive protein doesn't go through, but the radioactive DNA actually continues onward into the next generation. So it has been shown then that the material that transfers the genetic information, it is not protein, it is DNA. So what it is this is a very famous experiment, and so you should know about that one. All right, so what is the structure of DNA? DNA is composed of 4 different types of nucleotides. These are like the building blocks of DNA. And so DNA is a long chain, and it's made up of these blocks, these building blocks, these bricks, if you will. Like if you had a brick house, each brick is making up the house. Well, each nucleotide is the basic unit that makes up the DNA. But the nucleotide has 3 parts, and here they are: the phosphate group, the deoxyribose, and the nitrogen-containing base. Right, and in this picture, here's the base, here's the sugar, and here's the phosphate. These 2 are going to stay the same, but the thing that makes DNA different is that you have 4 different nitrogen-containing bases. Sometimes we call them nitrogenous bases. And you already know a little bit about the story. There's the adenine and thymine, which always hook up to each other. And there's cytosine and guanine, which also pair up with each other. And there it is right there. There's adenine and thymine, and there's cytosine and guanine. And those are the 4 base pairs in DNA. So you can see a little bit more about their structure right there, AT and CG. Those make up the steps of the ladder. If you can think of DNA like a ladder is twisted, we call it a double helix. The sugar and the phosphate that make up the sides of your DNA, but the steps of the ladder are made up of the nitrogen bases. Here are the guys that figured it out. These are Watson and Crick. Dr. Crick is on the right. He was from England. And Dr. Watson is down on the left of this picture. And I actually met Dr. Crick many times. He came to Los Angeles and was able to share with us some of his insight, and then we met him afterwards. So pretty cool deal. All right, so Watson and Crick. Let's go on. And the thing is, the thing about this story that's tragic is that Watson and Crick didn't do the actual experiments. They just took the information from other people's experiments and they figured out the structure of DNA. So what really pushed them over the edge and helped them figure it out is the Chargaff's information that said that you have A pairs with T and C pairs with G, but percentages are the same. Like if you had 20% A, you're going to have 20% T. And if you have 30% C, then you have 30% G. So that's how they figured out that these guys match up, make up the steps of the ladder. But then what about the structure? That kind of, who would have thunk that? Well, what they did was they took Rosalind Franklin's X-ray images, and she had all this information, and they took this information, they figured out that DNA then is a double helix. Why didn't she win the Nobel Prize? She actually got sick from all the experiments that she did with X-rays. So she actually died before the Nobel Prize was awarded. So since they don't give the Nobel Prize to people who are dead, she didn't get a Nobel Prize. Although in history now, historically, we look at it, we go, oh, she should have got one. And she did much of the work that was important that led to the discovery of DNA. So again, A pairs with T and C pairs with G. It's a ladder that's twisted up. And so we call it the double helix. And if you know one side, then you know the other side. And the steps of the ladder are held together by hydrogen bonds, which, if you remember from chemistry, are the weaker bonds, not as strong as covalent or ionic bonds. And so those can easily be broken, but they can easily be put together as well. So we have AT and CG. All righty. And that's what I have here. Kind of got ahead of myself, but the bases are hydrogen bonds and the backbones are covalent. So these are strong and they're going to stay and they're stable. But these dotted lines here are transient and they can't be broken. But that makes it so that these guys the DNA molecules can unwind and unzip, come apart, and then come back together again. Righty? Well, how do they replicate? Well, here's the thing. They thought that that a certain way occurred, but actually experimental data showed us something else. So let's take a look at that. A single strand of DNA serves as a template for a new strand. That means that if you can make these guys come apart from each other, then the DNA is exposed and then both sides can be copied. So say you're a single strand, not the double strand where it's usually in. The single strand of DNA serves as a template, and the rules of base pairing direct the replication. This happens during mitosis, and actually it happens during the cell cycle before mitosis. And one of the steps of interphase, which is part of the cell cycle, is called the synthesis or S phase or S stage. And that's when the DNA is replicated before the chromosomes are separated out in the process of mitosis. So each body cell gets a complete set of identical DNA, and it's identical because you took the DNA and they came apart, and then you just copied the exposed sides. And here's a graphic that shows you the DNA is copied in this stage right here called the synthesis stage. It's proteins that do all this work. It's the DNA serves as a template, but the proteins and enzymes that do the work of replication. Enzymes unwind and unzip the double helix and hold it apart. And then the free-floating nucleotides can come in, but they still need a protein and enzyme to come in and glue them on there in essence. And so that is what's happening right here. You can see this. Here are the nucleotides that are free floating in a nucleus, and they come around and they are over here. You can see it's already partially done. See that these will go in and add to that exposed blue strand. And here are the enzymes that actually do the work. These are called DNA polymerase. If you don't remember from chemistry or you don't know yet, ASE means that it's an enzyme. And so this is an enzyme that creates a polymer, a big long molecule known as DNA. So this name means a lot. And you can tell from the name that it's an enzyme, and you tell what it does. Makes a long molecule, and it works on DNA. So DNA polymerase does that. The polymerase enzymes form covalent bonds, and these are the ones we expect to see that connect the edges or the steps of the ladder. When all this is done, you get two new molecules of DNA, and each has one original strand and a newly formed strand. So we call this a semiconservative method of DNA replication. But they didn't always think that this was the way it was. And so there are three different possibilities. And so they did an experiment called they being Metzelson and Stahl. Metzelson and Stahl did this experiment. So obviously it's called the Metzelson-Stahl experiment. And what they did was they grew this DNA in a heavy nitrogen solution. And then they took it out and kept going with this experiment. So the new DNA was not heavy nitrogen, but it was normal nitrogen. So that little difference was enough so that you could tell what was old and what was new. In this picture, the old DNA is gray. And so what's going to happen is this DNA is going to split, and the DNA then will be copied. Well, that's what happens right here. But over here, the DNA didn't split, it just gets copied. And over here, the DNA is not really getting split. It's just you're putting new parts in between. And so these two on the edge are not the way. If you take a look at the second generation after this one, it's even more apparent, and you can tell by looking at the banding patterns what is happening with with the heavy DNA and the light DNA, with the heavy nitrogen and light nitrogen. And here's that information. If you took a look at a band, and what you could do is grow it and then put it in a test tube, and then take the test tube and put it in a centrifuge. What the centrifuge does is it spins it around so many times per minute, maybe 1,500, 15,000 times per minute, very, 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 very quickly. And what that does is you have a solution that changes its density, like you have right here. This one, the solution is called cesium chloride. Right there in the cesium chloride test tube, where it's more dense here at the bottom and less dense up here, the DNA from E. coli, which is bacterium, then it's going to leave bands. And so at the first, when you first start the experiment, there's only going to be one band. It's right here because they've all been grown in that heavy nitrogen 15N. But then when you take it out of the heavy nitrogen, put in a normal solution, and you grow it, well, look, they all have a band at with it's half old and half new. And if we go back, the only time you have half old and half new is like these guys are both half old and half new, half old and half new. But this is half old and half new, and this is half old and half new. So this also might be the possibility if we look at the data. This then would result in two bands because this would be an old band and this is a new band. So we go back and look at the data. If that were the case, the one on the left, then there would be a band here and a band here. So that's why we could eliminate this possibility, the conservative possibility right here. But now we have to look out for the dispersive. Well, if this continued in a dispersive manner, that you would have one band all the time. But over here, what you're going to see is that you always have this, you always have one strand being conserved. So in the next generation, these two are going to split, and these two are going to split. And then the four single-stranded copies are going to have added to it a new strand. So if we go to the data, that means that two of them are going to be half and half, but two of them are going to be completely new. So is that what happened in the next generation? That's exactly what happened. And 2 out of 4 were completely new, 2 out of 4 were half and half. But if we go back over here, look at this guy. Well, if this is the way it is, then really you should have 4 bands. No, these guys will split up and these guys will split up. And if the new DNA gets added on here to each of the single strands that happens after you split this down the middle like that, well, then it's going to still be dispersive replication of DNA, and it still should be, and it should be one band and not 2. So that's not what we saw. So we were able to eliminate that possibility because you expect to have one band that starts getting closer and closer and closer to the orange N14 line, which is the normal nitrogen. 14 and 15, by the way, those are numbers you get by taking the number of proteins, sorry, not proteins, but protons and neutrons. And a nitrogen atom has 7 protons and 7 neutrons in the most common isotope. But the heavy one is called heavy because it has an extra neutron. So it has 7 protons and then 8 neutrons. So that little bit of difference makes this a little bit heavier and makes it have a band a little bit closer to the end. All right, so that's how we figure that out. So DNA replicates, as we saw back here. DNA replicates in what's called a semi-conservative manner. All right. So it happens at not just at one point, but it happens at many points. So it happens very quickly. And so in this picture, it happens at these 3 points right there. And all these 3 points will start expanding and come together. So that's how it can happen so quickly. And it's not just 3 points, it's many, many, hard to imagine. We can't even imagine how many points there might be. We also know we know much about the different enzymes and proteins that are necessary in order to make this chemical reaction go, the replication of DNA. So you can see in this graphic, it shows you all of that stuff. But to give you a little bit more detail, what happens is all of these different things happen. The first one is a DNA helicase separates the DNA, separates it so that the DNA helix, the twisted part of the DNA is no longer in existence that way, and it's straight up unwound. So it's easier to get to. And this happens also because topoisomerase breaks and reforms the DNA. So it doesn't keep spinning. There are single strand binding proteins right here that hold it open and prevent the whole thing from, although you've untwisted it, it has a tendency to twist back into its helix. These single-stranded binding proteins bind where the DNA is single-stranded and kind of hold it in that orientation. Then there's an RNA primer, and the primer makes it so that the enzymes can stick to it. So it says the primase synthesizes an RNA primer. And once you have the RNA primer, then the DNA polymerase can hop on there because now it has a place where it can land. And you can see that in the picture right here. And so what's going to happen is you can start moving from the left to the right, like there. Okay, 5 minutes. And then you have the leading strand of DNA. The DNA is synthesized continuously, and right here, the DNA is made straight up, but this DNA is going to open up going to the left in this picture, but DNA can only be read in one direction. So what has to happen is DNA is read by the DNA polymerase this way, and you can see there's a fragment right there, and another one's starting to be made there. So you need, because this one can be replicated continuously going that way, this one is made going this way, but you're going to have little gaps right there. So you have to close those gaps, and those fragments are called the Kawasaki fragments, but those fragments don't get glued together, and then you have 2 copies of your DNA. The chemical that does that is called DNA ligase, and I think you can think of it as glue, so DNA ligase. All right, so that is something about a little bit more detail about DNA replication. Again, it's fast and it's accurate. It starts at many points, as we saw already. DNA polymerase can also, at the bottom, you can see, and find and correct those errors. And that's what's happening right there in this graphic. You can see that the DNA in this graphic, and this should be, if you look at it, remember A goes with T and C goes with G. Here's A, this should be T. DNA polymerase is knocking this G off and put a T back behind that. All right, this is called mismatch repair. And it says here the incorrectly added base is detected after replication and not before. The mismatch repair proteins detect this base and remove it from the newly synthesized strand by nuclease action. And the gap is now filled with the correctly paired base. Oh, that's you may hear from in the news every so often. This is it's called the T, as you know, is thymine. We have thymine dimers that sometimes form. So as your nucleotide excision repairs thymine dimers, expose to UV, thymines align adjacent to each other and form thymine dimers in normal cell. They're excised and replaced because this is not normal here. So when they start to interact with each other, that is not good. So you can do that type of repair right there. I mean, dimers may cause different characteristics in people, and so here's one, Xeroderma pigmentosa. And so here are different types of mutations. Mutations can lead to changes in the protein sequence encoded by DNA. And you can see there are different types of mutations. Different types of point mutations. So here's silent, missense, nonsense. And what this is in a diagram is if you look at the DNA sequence, they're slightly different. So here's AGC, GTA, AGC, GT. This is incorrect. So that would be a problem, possibly, if this were an A or a T. But if you look down here, the amino acid that comes in is actually the same for GTA as GTT. So that didn't cause any problems. So yes, it's a mutation because of the change in DNA, but it did not cause a difference in the proteins. If you compare these here, though, here's valine, here's proline and tyrosine right there. They are different amino acids. So this is this is a mutation, a change in the DNA that caused a difference in the protein in the sequence of the amino acids. So you only see a mutation if it becomes apparent when there's a change in the protein, not necessarily a change in DNA, because this change in DNA didn't do anything. Sometimes that it's just a change at one point, so it's called a point mutation, but sometimes if you have a deletion or an insertion, it could totally change the way these are read. So you have AGC, GTA. Over here, you have AGC, G. These two right here, these TAs got cut out. So GCC. So it's as if the T and the A got taken out and then everything moves over. Now it moves over two. And so when you move over two, now you have, if you look at the order of the amino acids, it's totally different amino acids now. So frame shift mutations are typically much worse than point mutations and can cause big problems in proteins that you won't have the correct amino acid sequence. That means they don't fold correctly and then they don't work. It happens. All right. Okay. All right. Well, that was DNA structure and something about replication.